So now that we've explored how the genetic message on DNA is conserved through replication and passed on to future generations, let's consider its role within the cell. We know that the DNA contains a sequence of nucleotides or nitrogenous bases on the stairs down the center, and that these bases constitute the genetic code. But how is this genetic code utilized by the cell? We know that DNA does not perform the cell processes itself. However, it is conveyed into RNA molecules, which then carry instructions for production of proteins at the ribosomes. This concept of genetic information flow is considered the central dogma of biology. In the central dogma, information transfers from DNA to RNA, a variety of different types, eventually to a protein. The master code of DNA is used to synthesize an RNA molecule in the process called transcription. So transcription is the process of making an RNA transcript of the DNA. We're basically transcribing from one language, the DNA language, to a new language, an RNA language. After we've made an RNA transcript, the information from RNA is then used at the ribosome in the process of translation, where the nucleotide code is translated into a protein. Now, we've considered this as the central dogma for many years. However, we've started to recognize that there are exceptions to the central dogma. Central dogma implies DNA to RNA to protein. However, we've already learned about some viruses, the retroviruses, that actually come in as an RNA and then have reverse transcriptase in order to produce DNA that can then be transcribed and translated into the proteins that that retrovirus needs. So, the central dogma, DNA to RNA to protein, there are exceptions. Sometimes we're going to see RNA to DNA to RNA to protein. Also, we've noticed that it's incomplete because not all RNAs end up becoming proteins. So some RNA molecules are used to regulate gene function. So we have a large group of regulatory RNAs that will be involved in both the process of transcription as well as regulating the DNA itself. Many of the genetic malfunctions that cause human diseases are found in these regulatory RNA segments. As you're probably aware, in the Human Genome Project, they established that about 98% of the human genome didn't code for anything. They called it junk at that point in time because we didn't understand. But now, as it turns out, all these additional sections of the genome code for regulatory RNAs, so they don't ever actually get expressed as a protein. Now, of course, we understand it's not junk at all. It's really important stuff. So back to the central dogma for just a moment. I have an analogy that works really well here. Transcription, it's kind of an odd analogy. It's a Food Network analogy. So in this analogy, the nucleus is like your computer, and the DNA is the food network in there. We need to get a recipe off of the Food Network. So rather than taking the whole computer into the kitchen and risking messing it up, we're just going to take a gene or a recipe from the Food Network and we're going to make a copy of it. We'll print it out and then we have that recipe on a piece of paper or a piece of messenger RNA in this case. Now you have that recipe on the piece of paper. You're going to take it out of your office where your computer is located and you'll take it over to the kitchen. In this analogy, the kitchen is the ribosome. The ribosome is the site where you're going to take all the instructions from the recipe and change it from words now into actual food. In a similar fashion, we're taking DNA nucleotides. We're transcribing them into RNA nucleotides. So it's a similar language, but it's a different form of that language. And then we're taking it to the ribosome, where we're going to translate it entirely into a new language, from nucleotide language into protein language or amino acid language. I don't know if the analogy helps, but try it out if you like. So now that we understand the central dogma, the question becomes, how do the gene structures actually lead to expression of traits in an individual? And what features of the expression cause organisms to be distinct from one another? In essence, we're saying, what is the correlation between the genes on the DNA in the protein structure. We know that each gene is a linear sequence of nucleotides and that it codes for a protein, but how does it do this? The key here is in looking at the language of DNA, or the genetic code. And it turns out there's a triplet code relationship between DNA and proteins. That is, that 3 consecutive bases on a DNA strand, called a triplet, codes for a particular amino acid. When we take 3 nucleotides at a time, each nucleotide will code for a similar triplicate nucleotide sequence on the messenger RNA, which is a copy of one strand of the DNA. And then that triplicate code will code specifically for one amino acid. Now each triplicate code is slightly different and thus results in translation into a different amino acid. So when the triplet code is transcribed and translated, it dictates the type and order of amino acids in a polypeptide. So now we can understand that one gene differs from another in the triplicate code that it creates, and thus the sequence of amino acids that it determines in the process of translation. Keep in mind that proteins ultimately determine the phenotype. Everything about an individual and its appearance is based on proteins. DNA is mainly a blueprint that tells a cell which kinds of proteins to make and which kind of RNAs to make, as well as how to make them. So before we move on, we should examine the major players in transcription and translation. There are many players. Mostly we're going to see messenger RNAs and transfer RNAs, as well as some regulatory RNAs, ribosomes, and some other types of enzymes. So RNAs are the tools in the cell's assembly line. RNA differs from DNA in that it's a single-stranded molecule, still with a helical form, but it contains uracil instead of thymine as one of the nitrogenous bases. And as you recall, thymine is one of the pyrimidines, and so uracil is also a pyrimidine and thus a single ring structure. The sugar in RNA is ribose, hence the name ribonucleic acids as opposed to deoxyribonucleic acids. There are many functional types of RNAs. Some of them are small regulatory pieces, and other ones are large structural RNAs. The only type of RNA that's translated into a protein molecule is the messenger RNA. Let's look a little more closely at messenger RNA. Again, recall that messenger RNA is the RNA that carries DNA's message to the ribosome. It's a transcript of the structural genes or other genes in the DNA, and it's synthesized by a process similar to the synthesis of the leading strand during DNA replication. We'll look at it in more detail here shortly. The message of this transcribed strand is later read as a series of triplets or codons. You can see these codons exhibited down here in the figure below. One codon is 3 nucleotides long or contains 3 nitrogenous bases. The next really important type of RNA is transfer RNA. Transfer RNA we'll see much more about during the translation process, so after we've got the RNA transcript. They usually are of uniform length, about 75 to 95 nucleotides long. And the molecule has sort of a cloverleaf structure, as you can see exhibited down here. That cloverleaf structure then folds into a complex 3D helix shape that you can see over on the right. The bottom loop of the cloverleaf expresses a triplet or an anticodon, and that anticodon designates the specificity of the tRNA and complements the RNA's codons. So here's the bottom loop, and then here are the 3 nitrogenous bases that denote the anticodon that's on a transfer RNA. This anticodon will eventually match up with a codon that's on messenger RNA. We'll see how this works later on. At the opposite end of that molecule, though, is a binding site for an amino acid. So the amino acid attached to this 3' end of the RNA molecule. Each transfer RNA carries a different amino acid, And that's designated by what the anticodon says down here. So for each of the 20 different amino acids, there's at least one specialized type of tRNA to carry it. Some amino acids have multiple tRNAs. Next, we should take a look at the ribosome. The ribosome is the mobile molecular factory for translation or protein formation. Remember that proteins are composed of polypeptides that are folded. So we're going to get a polypeptide or a sequence of amino acids at this building site or a ribosome. The prokaryotic ribosome, recall, is a 70S ribosome. It's composed of tightly packed RNA as well as proteins. The RNA component is a long polynucleotide molecule. And the interactions of proteins and the ribosomal RNA, or rRNA, create the two subunits of the ribosome that engage in final translation of the genetic code. Here you can see the ribosome with its large subunit and its small subunit. Inside the small subunit, we're now seeing a large piece of messenger RNA that's running through the active sites of the ribosome. Here's where transfer RNA comes in. Each of these transfer RNA molecules is bringing in a new amino acid to the growing polypeptide chain. We'll see how this process works later, but that's just a quick introduction to the ribosome for you. Large subunit, small subunit. You've got transfer RNAs as well as messenger RNAs pictured in this image. Now, let's take a look at the details of how transcription itself works. This figure should be a great reference point for you. In general, transcription is a fairly simple process. You'll see that it's very similar to the process of replication, except we're only transcribing a very short section as opposed to the whole chromosome that we see during replication. In the process of transcription, the DNA code is converted into an RNA copy, or if we're in the Food Network example, we're taking the recipe off our printer that we just printed from the computer. The key enzyme in this whole process is RNA polymerase. You'll be relieved here that that's pretty much the only enzyme we need to know, because RNA polymerase does all its own unwinding. Only one strand of the DNA contains meaningful information. That is the template strand. So the RNA polymerase will find what's called a promoter region. The promoter region is on the template strand. And that promoter region basically tells the polymerase to dock here. And again, it's very rich in A's and T's. Why would it be rich in A's and T's? Aha! The two hydrogen bonds that occur between A and T allow it to be unzipped much more easily. So RNA polymerase is going to unwind the DNA. And again, it's only going to read from 3' to 5', just like we saw with DNA polymerase. And so it's growing a new strand, again 5' to 3'. So nothing's really different here. So once the DNA polymerase has logged onto the promoter site and uncoiled the DNA, then it can move along the template strand and transcribe. That is, add RNA nucleotides to our growing messenger RNA molecule in the process of elongation. So here we have the template strand that's being read from 3' to 5'. The polymerase is moving in this direction. The new strand, the early mRNA transcript, you'll see the first end to pop out is 5'. The 3' end is over here, still being manufactured. So during elongation, we're simply adding RNA nucleotides to this messenger RNA transcript. The polymerase will continue moving along, unwinding DNA as it goes and rewinding it behind itself. As you can see here, growing the new messenger RNA strand. It will keep transcribing the DNA into messenger RNA until it reaches a termination site. Once it reaches a termination site, the messenger RNA transcript is released and it's now available for translation. So hopefully this all makes sense, But let's take a quick look at an animation to make sure we have a good understanding of the process.
A structural gene is made up of a sequence of bases in a DNA molecule consisting of a coding region with an upstream promoter and a terminator downstream of the coding region. The transcription of a gene into a messenger RNA sequence is initiated when RNA polymerase attaches to the promoter region and forms an open complex. RNA nucleotides are then polymerized from nucleotide triphosphates to make a messenger RNA. The messenger RNA sequence is elongated as the RNA polymerase moves down the DNA molecule until the RNA polymerase reaches the terminator region. Transcription is terminated when sequences in the terminator region are encountered. This causes RNA polymerase to dissociate from the DNA molecule, and the completed transcript is released.